Section 8.4, Transcription. RNA carries DNA's instructions. Information flows in one direction, from DNA in the nucleus to RNA, which is made from DNA in the nucleus, to proteins, which are made in the cytoplasm. This idea is called central dogma. That's the flow of information from DNA to RNA to proteins. Replication copies DNA. This happens during S phase of mitosis. Transcription converts the message from DNA into an RNA message in the nucleus, and then this RNA leaves the nucleus to the cytoplasm, where translation interprets this DNA message that is now on the messenger RNA into a string of amino acids called protein. In prokaryotes, replication, transcription, and translation occur all in the cytoplasm at roughly the same time. Now, a prokaryote cell is much less complex than a eukaryotic cell, so these processes can happen quicker, as the life cycle of a prokaryote is much quicker than a eukaryote. In eukaryotes, processes are separated in location and in time. Replication and transcription happen in the nucleus. Remember, transcription is the forming of RNA, and this RNA that's formed from transcription is processed before it leaves the nucleus. We'll talk about that later on. Translation, which is where the actual making of the protein occurs, happens in the cytoplasm. Okay, so in this is nucleus, so DNA is being transcribed into messenger RNA. This messenger RNA gets processed where it is acted on to make it much more easy to get the code out, and then it leaves the nucleus into the cytoplasm, where it's shown into a ribosome to form a protein. Examples when gene expression occurs. Remember, gene expression would be when a protein that is coded for on the gene located on DNA and transcribed into RNA and then sent from the nucleus to the cytoplasm, to the ribosomes to make the protein. That would be the actual gene expression. So whatever that protein is, it's going to get made once that gene goes through transcription and then translation and then actual making of the protein. Examples of this would be the Arctic fox fur color changes, right? In the winter, it looks like this, and in the late spring, summer, fall, it looks like that, and it changes the fur color to blend in with its environment. In the wintertime, it's not making the pigment proteins that produce the fur color like you would have in this fox. And in the late spring, summer, and early fall, the melanin is produced, so it makes the fur color so it blends in with its environment better. The start of puberty, the proteins that are necessary for the formation of these secondary sex characteristics, increase in muscle mass, increase in genitalia, and all the other associated secondary sex characteristics that come on through puberty are controlled by expression of proteins. And another example would be healing of a cut. Okay, you get a scrape here, this girl Charles, she got a scrape. So your body's going to produce proteins that will help with the clotting to kind of make a temporary barrier called the scab, and then the new skin will grow under it to fill in the hole in the skin, and she'll be cured, healed. Okay, differences between DNA and RNA. Okay, the first thing we notice the difference between RNA and DNA is in the name. RNA is ribonucleic acid or ribonucleic acid because the sugar in there is ribose. DNA is deoxyribonucleic acid, so its sugar is deoxyribose. They both have this sugar phosphate backbone. Okay, this is the sugar phosphate backbone. Same thing here, sugar phosphate backbone. The difference being DNA is this double helix, meaning it's paired up. The bases are paired up in the middle. So this is double helix. and they still have, the nucleotide is still the sugar, phosphate, and base. It's just the sugar's different and the bases are not entirely the same. RNA is the link between DNA in the nucleus and protein synthesis, which takes place in the cytoplasm. Okay, so both DNA and RNA are composed of nucleotides. Nucleotides are a phosphate, a sugar, and one of 4 nitrogen-containing bases, so they're both the same, right? Both these guys, DNA and RNA, are made up of chains of nucleotides. The sugar for DNA is deoxyribose, and the sugar for RNA is ribose. In DNA, the bases are adenine, thymine, guanine, and cytosine, okay? But in RNA, thymine is replaced by this other nitrogen-containing base called uracil. Okay, so the bases in RNA are adenine, uracil, guanine, and cytosine. DNA is a double-stranded molecule because the bases are going to form hydrogen bonds between the one strand and the other strand according to base pairing rules. And RNA is a single strand of nucleotides, which is formed by these same base pairing rules, but is a complementary strand to the DNA from which it's made. Okay, so as you can see, the bases here, adenine, uracil, cytosine, and guanine, those would be these bases here on this single strand of RNA. Adenine, thymine, cytosine, and guanine would be the paired up bases that form the rungs of the ladder in this double helix model of DNA. So base pairing rules for DNA are adenine bonds with thymine, and thymine bonds with adenine, forming 2 hydrogen bonds. Guanine bonds with cytosine, forming 3 hydrogen bonds, and cytosine bonds with guanine, again with 3 hydrogen bonds. And RNA, remember, uracil replaces thymine, so uracil would bond with adenine, 2 hydrogen bonds. Adenine would bond with uracil, 2 hydrogen bonds. Cytosine would bond with guanine, 3 hydrogen bonds, and guanine would bond with cytosine, 3 hydrogen bonds. So there are 3 different types of RNA. The first, called messenger RNA, or mRNA, this is what's made during transcription. It's complementary to the DNA strand from which it's being read. If the DNA sequence was ATG, CCT, and TAG, and CTA, the messenger RNA complement that would form from this would be UAC, GGA, AUC, and GAU. Okay, this is messenger RNA. Ribosomal RNA, or rRNA, is what forms part of the ribosomes. Remember, ribosomes are the organelle in the cytoplasm, either floating freely in the cytoplasm or bound to rough endoplasmic reticulum where protein synthesis takes place. This is where proteins are made. Transfer RNA, or tRNA, brings amino acids from the cytoplasm to a ribosome to make the growing protein. So all 3 of these types of RNA are necessary for protein synthesis or gene expression. They just all have different jobs, different parts to this whole process of gene expression. Okay, with the 3 different types of RNA, they also have 3 different shapes. This would be messenger RNA, which is linear, just a long chain. Right? Ribosomal RNA is actually 2 loops of RNA that compose the ribosome and other proteins in there. And then tRNA, or transfer RNA, has this shape where it's going to have kind of like a T type thingy, and it's got these 3 bases on the end, which are what we're going to talk about in a minute. But those things are going to bind with the 3 bases on the messenger RNA to bring in the appropriate or the correct amino acid for the growing protein chain. So transcription is a process of copying a sequence of DNA, which would be a gene, to produce a complementary strand of RNA. Remember, complementary means it would be the RNA complement according to base pairing rules. Adenine to uracil, thymine to adenine, guanine to cytosine, cytosine to guanine. Those complement strands, when this is catalyzed by an enzyme called RNA polymerase, okay, this bonds the nucleotides together in a chain to form RNA. Okay, so you can see from this is a DNA molecule that's opened up. Okay, so there's enzymes that opens this up, and then this enzyme, RNA polymerase, starts adding bases according to the complementary ones here. Okay, as you can see, this is happening. These guys form complement all the way down, and so we get this strand of messenger RNA that was read from the unzipped or open double helix of DNA. So transcription, again, is writing of RNA from DNA. The RNA polymerase recognizes a start of a gene and unwinds a segment of the DNA molecules. Remember, it's not going to open up the entire molecule because it only needs this one small part, this one gene. Okay, the RNA polymerase strings or attaches complementary RNA nucleotides together using the template. The template would be part of the DNA molecule that was opened up, and it would be reading those bases and bringing in the appropriate RNA nucleotide. Again, these are the RNA base pairing rules. Okay, and then third, the RNA strand detaches from the DNA. Before it can leave the nucleus, it has to be processed. We'll talk about this processing later, but it has to be processed in order to get it like pared down, so it's containing just the information for the gene that is to be expressed. So transcription process is similar to replication. Okay, so similarities are this. It occurs in the nucleus. It's catalyzed by large enzymes. In order for it to happen, the DNA double helix has to be unwound. It follows base pairing rules or complementary base pairing rules, and because of the enzymes and the checks that go along with it, it's highly regulated. Okay, the difference is replication is to make DNA before a cell divides, so each new cell after mitosis will have one complete set of DNA identical to the cell it came from. And this only happens once during each round of the cell cycle. Transcription, on the other hand, since we're only doing a small part of the DNA molecule, this can occur thousands of times. The same gene can be transcribed thousands of times, and then many RNA molecules can be transcribed from a single gene, because there are times when you're going to need a whole bunch of proteins at once. So if you can make more messenger RNA molecules that will code for that protein, you're going to get a lot more proteins made when you need them.